Hello guys, myself Sudha studying in AIIMS Nagpur. Let's see how many guys had revised biomolecules after first session. See, let's revise some important concepts which had discussed earlier. What are biomolecules? See, biomolecules include four different categories. One is carbohydrates, another is proteins and fat, etc. See, in carbohydrates, it includes monosaccharides, disaccharides, and polysaccharides. See, when monosaccharides these are linked together with the help of glycoacidic bond, it forms polysaccharides. See, for example, glucose is a monosaccharide. When glucose are linked together with the help of glycoacidic bond, it forms polysaccharide. So polysaccharide is cellulose. See, and fructose is a monosaccharide. Polysaccharide of fructose is inulin. When glucose and fructose are linked together with the help of disaccharide bond, it forms disaccharide, which is sucrose. Sucrose is a disaccharide which is formed with the help of glucose and fructose. After seeing sucrose, we should remember that one concept about sucrose, that is inversion of sugar. What is meant by inversion of sugar? Sucrose is a dextrorotatory molecule with 62.5 degrees. When it hydrolyzes, it forms dextrorotatory glucose, which is 52.5 degrees, and levorotatory fructose, which is minus 90. So there is a conversion of sucrose molecule from dextrose into levose. The net rotation on the right side is minus 40. So there is a conversion of sucrose from dextrose to levose. It is known as inversion of sugar. And next important concept about proteins. Proteins these are exist in four forms. One is primary, next secondary, tertiary, and quaternary. In primary form, amino acids these are linked together with the help of peptide bond. What is meant by peptide bond? Peptide bond is the bond between carbolic acid of one amino acid and amine group of another amino acid. Peptide bond is formed by releasing water. It cannot be distorted by heat. So peptide bond is a strong bond which cannot be distorted by heat. Next, in secondary, tertiary, and quaternary structures, amino acids are linked by hydrogen bonds, which are very fragile. So we can easily break hydrogen bond. So denaturation of secondary, tertiary, and quaternary structures leads to distortion of structure. It causes distortion. So we all know that protein exists in four forms, which are primary, secondary, and tertiary and quaternary proteins. In primary structure, where amino acids are linked to each other with the help of peptide bond. Peptide bond is the bond that is between carbolic acid of one amino acid and amine group of another. So it is a strong bond which cannot be distorted by heat. And secondary, tertiary, quaternary structures, these are linked together by hydrogen bonds. We all know that hydrogen bonds, these are the fragile bonds, easily to break. So on denaturation of these structure, we can easily break the structure. Next, fats. We all know fats undergo beta oxidation to produce ATP. This is about fats. We all know in our body major energy source is glucose. Glucose undergoes aerobic metabolism in the presence of oxygen, anaerobic metabolism in the absence of oxygen. The first step of glucose oxidation is glycolysis. In glycolysis, glucose is oxidized to form pyruvate, ATP, NADH2, and water. Further, pyruvate, in the presence of oxygen, it undergoes Krebs cycle to yield more energy. In the absence of oxygen, it produces ethanol or lactic acid. This is about glucose oxidation. Without further delay, let's start our session of remaining questions. The question is the reaction of A, oxygen, and B, carbon monoxide with heme. We all know that heme, it is a protein in quaternary form. It contains 2 alpha subunits, 2 beta subunits. Heme is a carrier of oxygen molecule. It carries oxygen to deliver to tissues and CO2 to lungs. So this is the function of heme. They are asking what is the reaction between heme and carbon monoxide and oxygen. We all know when heme reacts with oxygen, it forms oxyhemoglobin. When heme reacts with carbon monoxide, it forms carbon monoxide hemoglobin. So we all know that heme has more affinity towards carbon monoxide. It has more affinity towards carbon monoxide. The bond is more stable. So affinity is also increased towards carbon monoxide. Affinity of heme with carbon monoxide is greater than comparatively to oxygen. So bond stable in HbCO is more as compared to HbO2. So option will be D, C. Both oxygen, heme and carbon monoxide complex, but carbon monoxide with heme complex is more stable because affinity of carbon monoxide towards heme is more as compared to oxygen. Next question. Identify the element amongst the following which is responsible for oxidation of water to O2 in biological processes. What is the oxidation of water? When 2H2O undergo oxidation, it produces O2 plus H2. There is a loss of electrons in case of oxygen. In oxidation state of oxygen in water is -2. When it undergoes oxidation, it produces oxygen, where oxidation state is 0. So oxygen loses electrons. When oxygen loses electrons to convert from water to oxygen. So they're asking which element has the capacity to oxidize oxygen. So option, in case of manganese, we all know the electronic configuration of manganese is 1s2, 2s2, 2p6, 3s2, 3p6, 4s2, 3d5. It has 5 unpaired electrons in d orbit. So it has more oxidizing power as compared to others. So oxidation of water to oxygen is done by manganese because of these 5 unpaired electrons, which contribute to more oxidizing power of manganese. C. Because of these 5 unpaired electrons, it can convert oxygen in hydrogen to oxygen molecule. So the best option is B. Next question. In reference to biological roles, calcium ions are important in C. First option, they gave triggering the contraction of molecule. Second option, generating right electrical potential across cell membrane. Third, hydrolysis of ATP. Fourth, defense mechanism. We all know that for contracting muscles, we need calcium to bind with actin-myosin complex. Then only when calcium binds with actin and myosin complex, our muscle contracts. Without this calcium, our muscle cannot contract. So contraction of muscle needs calcium. Next, option B, generating right electrical potential across cell membrane. We all know that electrical potential is maintained across cell membrane by sodium and potassium. In intracellular fluid, the value of potassium is higher as compared to sodium. In extracellular fluid, sodium value is high. So these two ions maintain electrical potential across the membrane. So second option is incorrect. Next, hydrolysis of ATP. When hydrolysis of ATP requires magnesium as cofactor, not calcium. So option C is incorrect. Next, defense mechanism. We all know that in our body, defense mechanism is maintained by immune system. So in immune system, whenever there is foreign antigen, it is reacted with antibody and form inactive complex. So in our body, defense mechanism is maintained by immune system, where it produces antibodies which react with foreign antigens and form the inactive complex. So the correct option is option A. Next question. Which of the following protein destroys the antigen when it enters in the body cell? See, here only we discussed about defense mechanism in our body. In our body, whenever we encounter foreign antigen, our body senses this and it releases antibody which is specific to this antigen. It will be specific. Antigen will be specific to the antibody. So then antibody reacts with antigen which makes it inactive by forming inactive complex. See, this is our defense mechanism. Whenever our body sends foreign antigen, it releases antibody in a specific manner which is specific to this antigen only. Though it combines with antigen and forms antigen antibody complexes and makes an antigen inactive. So option will be A. Option B is chromoprotein. Chromoprotein is the one which gives pigmentation to the tissue. Chromoprotein is a type of protein which gives pigmentation to the tissues. Next, option C, insulin. We all know that in our body, blood glucose is maintained by insulin and glucagon. Insulin, it acts whenever blood glucose levels are high. Glucagon, whenever blood glucose levels are low, it acts and increases the blood glucose level. These two hormones maintains our blood glucose level. Option D, phosphoprotein. Phosphoprotein, it is the protein. See, we all know that in ATP we have phosphate bonds. This is the phosphoprotein present in the ATP. Whenever breaking of this bond, see, whenever ATP, it contains 3 phosphate bonds. Whenever one bond is broken, it produces ADP and some energy. So, whenever phosphoprotein is metabolized, it produces energy. We all know that ATP contains 3 phosphate bonds. See, after hydrolysis of one phosphate bond, it produces ATP. After hydrolysis of one phosphate from ADP, it produces AMP, adenosine triphosphate to adenosine diphosphate to adenosine monophosphate. This is the option will be antibodies. Next question. The correct statement regarding DNA and RNA. We all know that DNA consists of nucleotide, nitrogen base, and sugar. And RNA consists of nucleoside, nitrogen base, and sugar. The sugar between DNA and RNA are different. DNA of sugar will be 2-deoxyribose and DNA of sugar of RNA will be ribose. So we all know that the nucleotide is made up of nucleic acid phosphate group, and nitrogen bases of DNA includes adenine of DNA, adenine, thymine, guanine, and cytosine. If nitrogen bases of RNA includes adenine, uracil, guanine, and cytosine. C. DNA consists of nucleotide, nitrogen base, and sugar. Then sugar of DNA is 2-deoxyribose. And the sugar of RNA is ribose. So they are asking about what is the correct statement. The option A is the sugar component in RNA is arabinose, which is wrong. The sugar component in DNA is 2-deoxyribose, which is correct. Option B is the sugar component in RNA is ribose and the sugar component in DNA is 2-oxyribose. So option B is the correct option. Next question. In DNA, the linkage between nitrogenous bases. We all know that the nitrogenous bases in DNA are adenine, thymine, guanine, and cytosine. How they are linked to each other? We all know DNA is a double helical structure. Double helical structure means it contains two strands. So one strand it contains A, it pairs with thymine with the help of 2 hydrogen bonds. If in one strand it contains guanine, it pairs with cytosine with the help of 3 hydrogen bonds. And if thymine is present on one strand, it pairs with adenine with help of 2 nitrogen bonds. If cytosine in one strand, it pairs with guanine with the help of 3 nitrogen bonds. So these are the nitrogenous bases of DNA. So they are linked to each other from one strand to another strand with the help of hydrogen bonds. They are asking how the linkage between different nitrogen bases are. The linkage is hydrogen bonding. So the correct option is C. Next question. Which one of the following statement is incorrect about the enzyme catalysis? See, option A, enzymes are mostly proteinaceous in nature. We all know that enzymes are the substances which are made up of proteins. So option A is correct. Next, option B, enzyme action is specific. See, enzyme has some cleft which is specific to the substrate. See, this has some pocket which only this substrate can react with this enzyme. So enzymes are made in such a manner where one enzyme can react with one substrate only. So enzymes are specific. It forms enzyme substrate complex. So enzymes are specific in nature. Next, enzymes are denatured by UV rays and at high temperature. See, enzymes are made up of proteins. Proteins can be denatured and by temperature and by UV rays. See, proteins can easily be disturbed at secondary, tertiary, and quaternary structures. Enzymes, these are present in tertiary structure, so it can be easily denatured with the help of temperature and UV rays. So option C is also correct. Next, option D. Enzymes are least reactive at optimum temperature. See, this is wrong. We all know that this is the graph between enzyme and temperature. This is the optimum temperature where enzyme action is maximum. And at low temperature, enzyme action is very low. Even at high temperature, enzyme action is low. Except at optimum temperature, where enzyme can function maximum. See, this is the temperature where enzyme can work efficiently. This temperature is known as optimum temperature. Even at low and high temperature, the function of enzyme is minimum. So option D is incorrect. Next question. The segment of DNA which acts as the instrumental manual for the synthesis of protein. See, we all know from DNA we produce RNA. From RNA, proteins are synthesized. In DNA, we have nucleotides, which is made up of nucleic acid, nitrogen base, and sugar. Nucleotide is made up of nucleic acid, nitrogen base, and sugar. When it converts into, it forms nucleoside, nitrogen base, and sugar, which produce Protein. Proteins, these are linked by amino acid. Protein is made up of amino acid. When DNA is converted into RNA, RNA is synthesized protein. So what are the segment of DNA which acts as instrument manual for the synthesis of protein? DNA consists of nucleotides, which it is the segment for the synthesis of protein. These nucleotides converts to form nucleoside. With these nucleoside, which converts to produce protein, produce amino acids which are linked together to form protein. So the segment of the DNA is nucleotide, which is useful for the synthesis of protein. Next, in DNA, the complementary bases. We all know that DNA contains 4 nitrogen bases. It includes adenine, thymine, guanine, and cytosine. Adenine complements with thymine with the help of two nitrogen bonds, and guanine also complements with cytosine with the help of 3 hydrogen bonds. See, these are the complementary bases. Whenever thymine is present, it only bonds with adenine. This is meant by complementary system. See, adenine only combines with thymine, and thymine only combines with adenine. Here also, guanine only combines with cytosine, and cytosine only combines with guanine. These are known as complementary bases. So complementary bases are adenine and thymine, and guanine and cytosine. It will be option A. Next question. RNA and DNA are chiral molecules. Their chirality is due to C. Their chirality is due to mainly of sugars. DNA consists of 2 deoxyribose, and RNA consists of ribose. See, let's see the structure of deoxyribose and ribose. The structure includes deoxyribose contain 5 carbons. See, 5 carbons. These are linked in this manner, where first carbon include OH group and H group, where second carbon include H and OH, where third carbon include H, OH, and fourth carbon include CH2OH and H. What is meant by chirality? Where carbon consists of 4 valencies. So it can form 4 bonds. So if these bonds are attached to different groups, when these bonds are attached to different groups, that is known as chiral molecule. See, chirality, where one carbon is attached to the oxygen, first carbon is attached to the oxygen OH, H, and this second group. And second carbon is also a chiral, where one is attached to H, OH, and this is a different one. Third, where third carbon is attached to H, OH, and this one, which is a different one. So sugar makes the RNA and DNA are chiral molecule. Let's see the ribose structure. Ribose structure includes OH, H, 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 OH, and CH2OH and H. See, ribose molecule includes, this is also, it is a chiral substance, where first carbon is attached to CHOH, OH, and second carbon is also attached to HOH, third carbon HOH, fourth carbon CH2OH and OH. So it is a chiral molecule which makes the sugar makes the RNA and DNA with the help of sugar. Next one. During the process of digestion of proteins, the present in the food material are hydrolyzed to amino acids. The enzyme involved in the process. Proteins, with the help of enzyme A, it converts into polypeptides, and with the help of enzyme B, it converts into the amino acid. We all know in our body, protein digestion is mainly done by enzymes pepsin, chymotrypsin, and trypsin. See, These are the three enzymes which makes our protein to digest. In stomach, where pepsin acts on protein, converts into polypeptides. This is the reaction which is carried by enzyme pepsin. And polypeptides enter small intestine, which further converts into amino acid with the help of enzyme chymotrypsin and trypsin. See, trypsin and chymotrypsin, these are inactive enzymes. They need to be activated. So trypsin is activated from trypsinogen with the help of HCl, and it converts into trypsin. So trypsin, it activates chymotrypsin from chymotrypsinogen. So these trypsin and chymotrypsin, these two are inactive enzymes which need to be activated. So these are activated with the help of hydrogen chloride, which is acidic pH maintained by stomach. So these need to be activated, then only it can convert polypeptide into amino acids. Amino acids are the last substrates. These are the last substrates of protein. These are the last substrates of protein. So option will be B, pepsin and trypsin. C. In case of invertase, zymase, amylase, maltase, these are the enzymes required for breaking of sugars. See, maltase. This is the enzyme required to break maltose. This is the enzyme is used for breaking maltose, which is a disaccharide molecule. So after using maltase, it produces two monosaccharide substance. This is the enzyme used. Enzyme used is maltase. Next question. So option will be B. Next one. Which one of the following is not true regarding dextrorotatory lactose? On lactose is a disaccharide which is made up of glucose and galactose. These are the two monosaccharides which combine to form lactose. Let's see the structures of glucose and galactose. These are 6-carbon molecule. So glucose is a 6-carbon molecule. How these are formed? The bond between glucose and galactose is a bond between glucose and galactose is glycoacidic bond. These two are epimers. You know what is meant by epimers? Epimers is the substance completely same, except completely same. They differ at 1 carbon. So what is meant by epimers? Epimers are the substance which are completely same. The molecule of glucose and galactose, it is completely same, but they differ at 1 carbon. That carbon is 4th carbon. So these two are linked together with the help of glycoacidic bond, and these two are epimers which are different at 4th carbon, and these are completely same except at 4th. This is meant by epimers. So when lactose breaks down, it produces glucose and galactose. So they are asking which is not true. On hydrolysis of lactose, it gives equal amount of glucose and galactose. That is true. Galactose is a beta glycoside formed by the union of dextrorotatory glucose and dextrorotatory galactose. Second option is also a true. These two combines form lactose. Both three are dextrorotatory molecules. Next one. Lactose, option C. Lactose is a reducing sugar, does not exhibit mutarotation. See, this is wrong. What is meant by mutarotation? Mutarotation is the thing when light is passed to the sugar, passed down the sugar, it disperses light. This is known as mutarotation. So lactose exhibit mutarotation. So when light disperses on lactose, it diverges light. It is known as mutarotation. Lactose exhibit mutarotation. So option C is incorrect. Next, option D. The lactose consists of 8 OH group. This can be remembered with the help of structure of lactose. See, lactose is made up of glucose and galactose with the help of glycoacidic bond. So the structure will be this one. First will both are same but they differ at fourth carbon. So we have to write this is the first carbon, second, third, fourth, fifth, and sixth. So they differ only at fourth carbon. So let's see how they differ. This is the OH group, this is the H group, this is OH, this is CH2OH. In case of here, it is CH2OH, it is OH, this is OH, this is OH. See, they differ this position, OH and OH. They're asking how many groups are of OH present in lactose. See, 1, 2, 3, 4, 5, 6, 7, 8. So, with lactose contains 8 OH groups. So, option D is correct. Option C is incorrect. So, option will be C. Next question. A sequence of how many nucleotides in messenger RNA makes a codon for an amino acid? We all know that DNA is converted to RNA. And the process of conversion of DNA to RNA is known as transcription, where DNA is converted into RNA. When RNA is to synthesize protein, so RNA conversion of RNA into protein is known as translation. See, this is the process where messenger RNA is converted to protein. See, messenger RNA is readed by tRNA, which helps in the synthesis of protein. See, we all know that three bases combine to form one protein. This 3 bases is known as codon, and one codon can code one amino acid, and it is specific for each amino acid. But one amino acid can be coded by different codons. See, for example, we all know AUG. It is first codon which starts the process. We all know there are 3 terminator codons. 3 terminator codons which stops the process. So for the synthesis of one amino acid, we need 3 bases, that is codon. So 3 bases combine to form one codon, and one codon is specific for one amino acid. One codon is specific for one amino acid, but one amino acid can be coded by different codon. We have starting codon and ending codon. Starting codon is AUG, and the ending codon will contain 3. So option will be A. Next question. The enzyme which hydrolyzes triglycerides into fatty acids and glycerol is called. See, triglycerides, it is a lipid molecule. Lipid molecules, when it hydrolyzes, it produces fatty acids and glycerol. So it is done with the help of enzyme lipase. We all know what is the action of maltase. Maltase, it causes hydrolysis of molecule maltose, which is a disaccharide. When it hydrolyzes, it produces 2 monosaccharide molecules, which are glucose and glucose. Lipase, pepsin. Pepsin helps in breakdown of proteins. When proteins are break down with the help of pepsin, it produces polypeptides. And on further action of trypsin and chymotrypsin, it produces amino acids. So this is the action of pepsin. And what is the action of zymase? Zymase is the one which helps in carb metabolism. So option will be B. Lipase helps in the hydrolysis of triglycerides into fatty acids and glycerol. These are the end products of These are the end products of triglycerides. See, when triglycerides are hydrolyzed, it produces fatty acid and glycerol. These fatty acids and glycerol then cannot be metabolized further. These cannot be metabolized further. That's why they are called as end products. See, fatty acids and glycerol, these are the end products of triglycerides. So option will be B. Next question. The Chargaff rule states that in an organism, amount of bases all are equal. Option A. Option B. Amount of adenine is equal to that of thymine and amount of guanine is equal to that of cytosine. Option C. Amount of adenine is equal to that of guanine and amount of thymine is equal to that of cytosine. Option D. Amount of adenine is equal to that of cytosine and amount of thymine is equal to that of guanine. See, what is Chargaff rule? Chargaff rule is adenine to thymine equals to guanine to cytosine equals to 1. So amount of adenine is equal to that of amount of thymine, and amount of thymine is equal to that of adenine, and amount of guanine is equal to that of amount of cytosine. So then amount of bases are all equal. It is false. See, adenine and thymine, these are complementary bases. So the amount of adenine will be equal to that of thymine, but these two some value maybe is greater than that of guanine and cytosine. So amount of bases are not equal. Option B. Amount of adenine is equal to that of thymine is correct, and amount of guanine is equal to that of cytosine. Option B is correct because these adenine and thymine are complementary bases. What is meant by complementary bases? Adenine only bonds with thymine and thymine only bonds with adenine. And guanine and cytosine are also complementary bases. Guanine bonds with cytosine and cytosine only bonds with guanine. So option B is the correct option. Next question. Enzymes are made up of edible proteins with specific structure, nitrogen containing carbohydrates and carbohydrates. See, enzymes are not made up of edible proteins. See, edible proteins can be digestible. If enzymes are digested, so the process which enzyme need to be act is inhibited. So enzymes are not made up of edible proteins. We all know that enzyme have specific structure. This is enzyme. It has specific space where only specific substrate can react to form enzyme substrate complex. This is the enzyme substrate complex. See, this enzyme is specific for this substrate only. Enzyme has specific nature which only specific substrates can react. Next one, nitrogen containing carbohydrates and carbohydrates. See, and these enzymes need to be performed. When enzymes are made up of edible proteins and carbs, nitrogen containing carbohydrates, then these can be digested. So the function of enzyme is hampered. Enzyme function is hampered. So option B is the correct option. See, this is the chapter of biomolecules. This is the PYQ of biomolecules. We all know that biomolecules is a very tricky chapter. You have to revise biomolecule. Biomolecule, it comes under inorganic chemistry. Inorganic chemistry. See, inorganic chemistry is just like bio. Bio, you always focus on bio because In biology, you always have to remember the facts, you have to memorize every line of NCRT. Biomolecules will also come under inorganic chemistry, where everything need to be remembered. Have to revise multiple times. Have to revise multiple times to achieve perfection in this chapter. You have to remember everything. Revise multiple times. See, chemistry consists of three parts: physical and inorganic and organic. See, it consists of physical chemistry, inorganic chemistry, and organic chemistry. Physical chemistry is just like problem solving things, where you should understand the concept, and it is a very easy topic. You don't you should not memorize everything. Only concept need to be understand. Where inorganic and organic, you have to facts remember the facts, which makes chemistry very scoring. So just like bio, most of the portion of chemistry is made up of inorganic and organic. Just like see, 2/3 part is memorizing one, and 1/3 is you have to solve the problems. So if you are perfect at this 2/3, which makes easy for you to score in chemistry. So just like bio, in bio you have to remember everything. You have to remember everything, every NCRT line. Then only you can score. See, the scoring subjects in NEET exam is bio, chemistry, then physics. This subject makes different from others. Physics makes you stand out of the crowd. So, how to study physics? How to study physics? Physics can be studied with the help of proper revision, proper techniques. See, if you are studying around 8 hours in a day, you have to at least spend 3 to 3.5 hours for physics. Then you have to practice everything in physics. First of all, you have to solve examples in physics. Then have to do NCRT questions in physics, NCRT concept and questions in physics, and you have to do modules problems. This makes you get perfect in exam. See, we first have to practice everything. First, you have to understand the concept which you have to study. Then you have to practice examples. Then you have to practice NCRT questions. We have to understand NCRT concept. In physics, everyone thinks of only problems, but it also have some theory questions too, which makes difficult for you to solve if you are remembering only formulas. Please just understand the concept, then you can solve theory questions. Then you can also solve the problems by applying easy formulas. So this is the method where you have to study physics. Next, in case of bio we have to read every line of NCRT. Then we have some PYQ questions where you have to solve bio, and then you have to solve some module questions. Then only you get perfect. Then only you will be perfect in exam. Without this knowledge of solving, you cannot. Without this, we cannot solve anything. See, from December, now we are in December 2024. Exam is in May 2025. Here around 6 to 7 months. So have properly have to plan accordingly. Then only you can solve subjects easily. First, we should not have any backlogs. We should not have any backlogs. Then then we should not have any backlogs. Then you should revise. Then you should revise the topics properly according to the weightage. See, weightage of biomolecules is around 4%. So that means we get around 3 to 4 questions in NEET exam. So we have to revise properly to get correct of these 3 to 4 questions. See. Let's have some summary about biomolecules. Biomolecules. Biomolecule consists of 4 substances. One is carbs, another is proteins, fat, and other substances. See, carbs. These are the main one which provide energy. Proteins. These help in building collagen structure in our body. How collagen synthesis takes place? For collagen synthesis, we require proline and serine amino acids. In the presence of these amino acids, it synthesizes collagen. So, collagen makes our body strong. Fat. Fat is an adipose tissue. Fat acts as an absorbent and it is a thermal insulator. See, how have you ever wondered how polar bear lives in snow? Because it contains fat, huge amount of subcutaneous fat. Subcutaneous fat. So fat can insulate our body. It can insulate our body. Fat also acts as shock absorbent. Shock absorbent. After every organ, we can see some fat. For example, after kidney, we can see perirenal fat. After stomach, we can see some mesentery. In case of stomach, it is a after in case of kidney, it is surrounded by perirenal fat. Next one, how fat produce energy. For example, in malnourished person, where it does not provided with carbs, then protein and fat comes into play for providing energy. How protein and fat provide energy? Fat, it undergo beta oxidation. Then it produces acetyl coenzyme. Acetyl coenzyme enters Krebs cycle. In Krebs cycle, it yields ATP. In case of proteins, how proteins produce energy? Proteins break down into amino acids, where amino acids produces glucose with the help of process gluconeogenesis. We all know what is meant by gluconeogenesis. Gluconeogenesis is production of glucose from amino acids and other substrates. And after producing glucose, it undergo glycolysis. After glycolysis, the pyruvate is the substrate which produced from glycolysis. It enters the Krebs cycle. So we all know glucose is the main energy source in our body. What is meant by glycolysis and glycogenolysis and glycogenesis and gluconeogenesis. See, these are confusing topics. What is meant by glycolysis? Glycolysis is breakdown of glycogen into glucose and it produces energy to the body. Why we all need energy? Why we all need glucose for our body? It is the main source which produce energy and we can do our daily activity. We all know for muscle contraction we need ATP. For function of brain we need glucose. So without glucose we cannot survive. And what is meant by glycogenolysis is breakdown of glucose into glycogen into glucose. What is glycogenesis? That is production of glucose into glycogen. When does this take place? It takes place whenever our blood glucose is high, and then only it takes place. Whenever our blood glucose is high, it acts with the help of hormone insulin and converts glucose into glycogen. It only takes place whenever blood glucose is high. What is gluconeogenesis? Whenever you are deprived of glucose, we need more glucose, but when person is not able to provide glucose through carbs, so we produce glucose from amino acids and lipids. This is known as gluconeogenesis. Next, what is glycolysis? Glycolysis, this production of ATP or energy from glucose. Glucose undergo oxidative phosphorylation to produce ATP. These are some basic concepts which you need to remember. These are the basic concepts which you need to remember about carbs. How sugar molecules are combined with the help of glycosidic bond. How amino acids are combined in primary structure, peptide bond. It is a strong bond. And secondly, tertiary structure. Secondary structure, it is alpha helix and plate. In helical structure, we all know that is C double bond O and NH share hydrogen bonds that is present between amino acid of single peptide chain. This is alpha helix. And tertiary, where protein is further folded into tertiary structure. It is a complex structure. In tertiary protein, we have amine peptide bond, hydrogen bond, disulfide bond between amino acids. Quaternary structure. For example of quaternary structure, heme. It contains 2 alpha subunits and 2 beta subunits. This is about proteins. What is meant by denaturation of proteins? Denaturation means breakdown of protein with which using with the help of breakdown of protein with the help of heat, high temperature, UV rays. Only primary structure will is intact after doing denaturation. Example of denaturation is curdling of milk. This is example of denaturation. See, next one. This is denaturation of protein. Lipids undergo beta oxidation to provide energy. This is the end. This is from my side. Thank you so much. Please revise the chapter after this session. Thank you so much.